Today, we're diving into one of the most incredible stories in all of biology. It's the story of how your body creates a perfect microscopic weapon. We're talking about antibodies, and we're going to follow the elite special ops cell that learns to build them from the ground up.
So let's just think about this for a second. It sounds kind of impossible, right? That for every new virus, every new bacteria you encounter, your body can engineer a totally custom-made defense? Well, the secret lies in the amazing journey of one single type of immune cell.
So here's the game plan. We're going to follow a B cell through 5 critical stages of its life. We'll see it start out as a totally naive recruit, then get its mission orders, go through this intense training montage, and finally become the commander of a massive antibody army. Let's get into it.
Okay, so our story starts in the bone marrow with our hero, a brand new, naive B cell. This little cell is a star player in what we call the adaptive immune system. That's the part of your defenses that actually learns and remembers specific enemies. And more specifically, it's part of the humoral response, which is all about fighting bad guys that are floating around in your body's fluids, like your blood.
So what exactly is a B cell? Well, you can think of it like a highly specialized security guard. It's born with the potential to recognize one and only one specific threat out of millions and millions of possibilities. Its entire life is basically dedicated to finding that one specific enemy and then figuring out how to obliterate it.
So, what's this new B cell packing for its mission? Well, its most important piece of gear is the B cell antigen receptor, or BCR for short. Think of it like a unique, custom-made key. Through this incredible genetic shuffling process, your body creates billions of different keys, but each B cell gets only one type. It also has these molecules called MHC class II, which we'll see are super important later for showing other cells what it's found. This incredible specialization from birth is really the secret sauce to the whole system.
All right, so how does our B cell find its 1 in a million target? It can't just wander around the body picking fights. No, it has to travel to a very specific place to get its mission briefing. And that place is the lymph node.
You see, your lymphatic system is kind of like a network of highways that's constantly collecting fluid, junk, and you guessed it, potential invaders from all your tissues. It funnels all of this stuff into your lymph nodes, which act as these bustling security checkpoints where all the immune cells hang out to inspect for threats.
Now, inside the lymph node, the real action happens in a structure called a germinal center. When our B cell finally bumps into the one specific invader it's designed for, it helps kickstart the formation of one of these. And you can just think of this as an intense boot camp where that one single B cell is about to be transformed into an entire army.
Okay, so our B cell has found its target antigen inside the lymph node, but it can't just charge into battle yet. Nope. To get fully activated, it needs a critical confirmation, a second opinion, if you will, from a wise old mentor cell.
So how does the B cell show everyone what it's found? Well, first, its receptor binds directly to the invader. Then it literally swallows it, chops it up into these little pieces called peptides, and then uses that special MHC class II molecule to display a piece on its surface. It's basically holding up a flag for all the other immune cells to see, yelling, "Hey, look what I found."
And this right here is a really key distinction in immunology. B cells are unique because their receptors can see an enemy's whole three-dimensional shape, like being able to recognize an entire tank. T cells, on the other hand, they can't do that. They can only see a tiny fragment of that tank after it's been taken apart and presented to them on a platter.
And the mentor cell that comes over to inspect this evidence is a specialist called the follicular helper T cell. Now, this T cell has also been warned about the same invasion, but it heard about it from a different source. So now it needs to confirm that the B cell has found the exact same enemy.
So when that follicular helper T cell recognizes the peptide the B cell is showing it, this critical interaction happens. It's a molecular handshake involving these proteins called CD40 and CD40L. And this handshake, along with a burst of chemical messengers called cytokines, you can think of those as short-range text messages between cells, gives the B cell the final definitive signal. The threat is real, power up, it is time to build the weapons.
And now we get to the most intense and honestly the coolest part of the B cell's life. Inside that germinal center boot camp, it's going to undergo 2 incredible high-speed upgrades to forge the absolute perfect weapon. The first upgrade is called somatic hypermutation. What happens here is the B cell starts making tiny random mistakes, mutations, in the part of the gene that makes its receptor.
It's like a weaponsmith making hundreds of slightly different keys, testing each one to see if it can fit the lock even better than the original one did.
This whole process of trial and error is called affinity maturation, and it's basically evolution on hyperspeed. The B cells that have mutations that make their receptors bind tighter to the invader get the strongest survival signals, so they multiply like crazy. The ones that bind worse, they're eliminated. Now, this is a high-risk, high-reward strategy, but the payoff is an elite squad of B cells with receptors that are an absolutely perfect match for the enemy.
Okay, upgrade number 2 is antibody isotope switching. So the targeting part of the weapon is now perfect, but what kind of damage does it need to do? Depending on those cytokine text messages it gets from the T cell, the B cell can literally switch out the tail end of its antibody. This completely changes its function without ever changing what it targets. It's like keeping the same guidance system, but swapping a small bomb for a bunker buster.
And with that, the training is complete. Our little B cell has now fully matured into a professional plasma cell. And it's not a soldier anymore. It's a dedicated weapons factory. That finely tuned receptor that was stuck on its surface, the cell now mass produces that exact design as a soluble, free-floating weapon called an antibody, pumping out thousands of them every single second. So what can this incredible arsenal actually do?
Well, that isotype switching process creates a really versatile arsenal. You've got IgM, which is this big molecule that acts as the first responder. Then there's IgG, which becomes the main workhorse in your blood for long-term protection. IgA gets secreted out to guard your gut and your lungs. You've got IgE, a specialist that fights parasites, but is also the main culprit behind allergies. And then there's IgD, which, well, it mostly hangs out on naive B cells, and its full function is still a bit of a mystery. Each one has the same perfect targeting system, just a different job to do.
But no matter what isotype they are, these antibodies really fight enemies in 3 main ways. They can physically get in the way of a virus or a toxin to stop it from working. That's neutralization. They can kick off a domino effect of proteins that poke holes in bacteria. We call that complement activation. And maybe most important of all, they can do something called opsonization. Opsonization is just incredibly powerful. The antibodies act like bright red flags, completely coating the invader and tagging it for destruction. This makes it an irresistible meal for big, devouring cells like macrophages. It also lets other assassins, like natural killer cells, lock on and destroy the tagged enemy directly. Basically, they turn a sneaky, hidden enemy into a glowing neon sign that says, "Destroy me." And this incredible, exquisite specificity, this ability to find and bind to one single target out of trillions, it isn't just a marvel of biology. It has become one of the most powerful tools in all of modern medicine.
You see, by mimicking this natural process, scientists can now create huge batches of identical or monoclonal antibodies designed to target almost anything you can think of, from a cancer cell to a protein that causes autoimmune disease. This has totally revolutionized medicine. Which leaves us with a final thought. If we can now forge these perfect biological weapons ourselves in a lab, what are the limits to what we can fight next?